Welcome back, students. In today's video's lecture, we are going to discuss cellular respiration, that is part 12 of your unit 2, cell as a unit of life. But before I start the main lecture, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of original creator, owner of these images. If you are the owner of any of the images used in this particular PPT, have any concern, any issue, kindly contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss about cellular respiration, mitochondrial electron transport chain, high energy bond and oxidative phosphorylation. So in last lecture, we have discussed about glycolysis, right? Glycolysis, right? So as you can see over here, this glucose, after eating some food, that food means mainly carbohydrate, get converted into glucose, and this glucose immediately cross the cell membrane and come inside the cytoplasm of the cell, where this glucose has been utilized for the production of energy, right? So in the cytoplasm, this glucose gets converted into two molecule of pyruvate, which is a 3-carbon molecule, and this glucose is a 6-carbon molecule through a pathway known as glycolysis. And during this pathway, net gain of ATP is not about it exactly 2 ATP. Total 4 ATP has been produced during this glycolysis, but already in previous initial steps, we have utilized 2 ATP molecules, so net gain of 2 ATP occur during glycolysis, and this whole process occur in the cytoplasm. This we have discussed in our previous video lecture. When this glucose convert into pyruvic acid, this pyruvate then enter into the mitochondria. This is mitochondria. These mitochondria is present in eukaryotic cells, right? So this this can cross the membrane of mitochondria and come inside the matrix part, right? And where this pyruvate get convert into a molecule known as acetyl coenzyme A, which is a 2-carbon molecule. Acetyl coenzyme A. So from this 3-carbon molecule, this one further convert into a 2-carbon molecule. This one is 3 to 2-carbon molecule. It will convert into acetyl coenzyme A. Acetyl coenzyme A. So how it will convert? It must release one carbon from itself as a carbon dioxide, right? So first glucose to pyruvic acid, okay, then pyruvic acid to acetyl coenzyme A. This is known as glycolysis. This particular step is known as link reaction, and this acetyl coenzyme A then enter into a cycle known as Krebs cycle or TCA cycle. As you can see over here, this pyruvate then convert into TCA cycle, tricarboxylic acid cycle. That's why, and this cycle is a main cycle which continuously follow its step inside the mitochondrial matrix, right? So this occur in mitochondria and this occur in cytoplasm. So that's why this process is known as link reaction, because it act as a link between two main cycles, right? So these are the steps, and after the generation of so many NADH and FAD during this TCA cycle, they further utilize in oxidative phosphorylation for the production of ATP, as you can see over here. Clear? So what are those three main steps? In cellular respiration, three main processes which involve in this ATP production. First is glycolysis, which occur in cytoplasm, right? Then TCA cycle, which occur in matrix of mitochondria, and then final production of ATP, which occur during this oxidative phosphorylation. So these are the three important steps of ATP production, right? During glycolysis, only partial oxidation occur because only a single bond has been broken down. That 6 carbon molecule, that is glucose, get converted into pyruvic acid, that is 3 carbon molecule. So partial oxidation occur during glycolysis. And similarly, tricarboxylic acid cycle that occur during mitochondrial matrix, total ATP production doesn't occur that process. Yes, NADH, FADH2 has been produced, but final production of ATP has been occurred during oxidative phosphorylation. Okay, so these three processes of ATP production are the main three processes: glycolysis, TCA cycle, and oxidative phosphorylation, in which these two, tricarboxylic acid cycle and oxidative phosphorylation, this tricarboxylic acid cycle, as you can see over here, and oxidative phosphorylation, these occur within mitochondria. You can see over here, this is the cytoplasm. So this glycolysis occur in cytoplasm, while these two occur in mitochondria, right? So electrons that are passed through electron transport chain, ETC, ultimately generate free energy capable of driving phosphorylation of ATP, phosphorylation of ADP. Okay, that actually continuous passing of electron through one complex to another, which we will discuss further in coming slides, occur during this inner membrane of mitochondria, right? Okay, now, so what is cellular respiration? Let's brush up again. The cellular respiration, during the process of cellular respiration, glucose is oxidized to carbon dioxide, water, and it will generate energy as ATP. This process is known as cellular respiration. Means glucose to carbon dioxide, water plus ATP. This process is known as cellular respiration, where glucose is oxidized. Okay, so and in this particular reaction, there are so many metabolic steps are involved. We have already discussed glycolysis step, right? And this is, as you can see in this particular picture, the generation of pyruvate occur. This is the last product or main product of glycolysis, right? So generation of pyruvate through the process of glycolysis is the first step of fermentation as well. One molecule of glucose makes 2 molecules of pyruvate or pyruvic acid and 2 molecules of ATP during glycolysis. We have discussed it in a very detailed way in our previous lecture, where this glucose get converted into pyruvate, glucose to glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-phosphate, and so on. This 6 carbon molecule get converted into 2 molecules of 3 carbon molecule, that is pyruvic acid. This is pyruvic acid, and here we have utilized 2 ATP molecule, right, and produce 4 ATP molecule, 2 over here, 2 over here, because 2 molecules of pyruvate. So net production or net gain, 2 molecule of ATP occur during this process. Right. So this is the first step, glycolysis. And then this pyruvate, which is formed by glycolytic catabolism of carbohydrate in the cytosol or cytoplasm, after it enter the mitochondrial matrix, undergo oxidative decarboxylation by a set of reaction catalyzed by pyruvic dehydrogenase. As you can see over here, this pyruvic acid is the last product of glycolysis. This get convert into acetyl coenzyme. This one is a 3-carbon molecule. This one is a 2-carbon molecule. So of course, 1 carbon dioxide molecule has been released. And this reaction has been catalyzed by the pyruvate dehydrogenase enzyme. We can use the term oxidative decarboxylation because carbon dioxide has been released. That's why, and it is an oxidative process. That's why we can also use the term oxidative decarboxylation for the same reaction. Magnesium ion act as a cofactor over here. Okay, and 1 molecule of NADH also generated. And this reaction act as a link reaction. Because this reaction, act as a link between glycolysis and Krebs cycle. Okay, so 1 molecule of pyruvate generate 1 molecule of NADH. So 2 molecule has been generated from 1 glucose. So total we can say 2 molecule of NADH has been generated during the metabolism of 1 glucose molecule. Right? And this acetyl coenzyme A then enter into TCA cycle, or we can say tricarboxylic acid cycle or Krebs cycle, after the name of the scientist Hans Krebs, who actually first illustrate this particular cycle. Clear? Now, so whether it is a TCA cycle or Krebs cycle or citric acid cycle, because the first product of this cycle is citric acid, which is a 6-carbon molecule. And this citric acid contain 3-carboxylic group. That's why we can also use the term tricarboxylic acid cycle, because it is having 3 carboxylic group. Okay, that's why whether it is a TCA cycle or Krebs cycle or citric acid cycle, all 3 are same. Okay, and it is the second stage of cellular respiration. First stage glycolysis, second stage TCA cycle. The 3 stage of process by which living cell break down organic fuel molecule in the presence of oxygen to harvest energy that need to grow and divide. This metabolic process occur in most plant, animal, fungus and bacteria. In all organisms except bacteria, TCA cycle is carried out in the matrix of the mitochondria, matrix of intracellular structure called mitochondria. Okay, now this is a TCA cycle or citric acid cycle, just now we have discussed. And this pyruvate has been generated from, this is the last product of glycolysis, right? And 6-carbon molecule glucose. Glucose is the main source of energy in glycolysis, which further convert into fructose and then fructose 1,6-phosphate and so on. And 1 molecule of glucose will generate 2 molecules of pyruvate. Right? And pyruvate or pyruvic acid is a 3-carbon molecule. And this pyruvate then convert into acetyl coenzyme A, which is a 2-carbon molecule, and just now we have discussed as this convert in 3 carbon to 2 carbon, so obviously 1 carbon dioxide or 1 molecule of carbon dioxide has been raised during this process, and in this process NADH has been generated and coenzyme A also act as a participating in this particular reaction. So this acetyl coenzyme A enter into this TCA cycle, and after combining oxaloacetic acid, it will produce its first product, that is citric acid. This is a 2-carbon molecule and this one is a 4-carbon molecule. So obviously it will generate 6-carbon molecule acid, that is citric acid. Okay, and in citric, and this is the first product, that's why we can use the term citric acid cycle. The citric acid contains 3 carboxylic group, that's why we can also use the term tricarboxylic acid cycle, right? And in this whole cycle, only and only this one, means alpha ketoglutric acid is the only type of molecule present in this particular cycle. This one is having 5 carbon in it. 5 carbon in it. Which molecule? Alpha ketoglutric acid. Alpha ketoglutric acid is only type of acid which is present in this particular cycle having 5 carbon in it. After this, all the type of acid, they all are 4 carbon molecule. Okay, and we have already discussed that citric acid is 6,1. So obviously for the formation of alpha ketoglutric acid, the citric acid must remove 1 carbon as a carbon dioxide over here, and on this step also 1 molecule of NADH has been produced. And as I told you, this one is the only molecule is having 5 carbon in it. So after this also 1 molecule of carbon dioxide has been released as a carbon dioxide, and next product, next to next, they all are 4 carbon molecule, as you can see over here. Succinic acid, malic acid, oxaloacetic acid, all are 4 carbon molecule. Okay, so just before the production of alpha ketoglutric acid and just after the production of alpha ketoglutric acid, one molecule of carbon dioxide has been released, and on the same step one molecule of NADH has been generated. Clear? And after this step 1 molecule of ATP or GTP also produced. Okay, so from 1 molecule of pyruvic acid or pyruvate, 2 molecules of carbon dioxide, 2 molecules of NADH, and 1 molecule of ATP or GTP has been produced. Not 2 but 3 molecule of NADH from 1 molecule of pyruvate during Krebs cycle. As the succinic acid then further convert into malic acid, and during this conversion 1 molecule of FADH2 again produce. Okay, and then this malic acid then convert into oxaloacetic acid, which is further a 4 carbon molecule. This oxaloacetic acid then further combined with this 2 molecule acetyl coenzyme A and then again produce citric acid, which is further a 6 carbon molecule acid. Clear? So this is a whole cycle, and this whole cycle removal of 2 molecule of carbon dioxide occur, production of 3 molecule of NADH, production of 1 molecule of FADH2, and production of 1 molecule of ATP occur. And as you know that NADH, 1 molecule of NADH is equal to 3 molecule of ATP and 1 molecule of FADH2. FADH2 is equal to 2 molecule of ATP. But these ATP molecule will, you will get these molecule during next electron transport chain type, not here. Okay, but yes, obviously 1 molecule of ATP has been generated over here, 3 molecule of NADH to 1 molecule of FADH has been produced during citric acid cycle, and removal of 2 carbon dioxide molecule has been occur during this Krebs cycle. Clear? Now, electron transport chain. And this electron transport chain is a process by which the NADH, FADH2, which has been generated during Krebs cycle, are oxidized and a proton gradient is formed. Okay, and oxidative phosphorylation is also a part of this electron transport chain. It is a process of making ATP by using proton gradient, which is generated by electron transport chain. Clear? So respiration by mitochondria, oxidation of the substrate is coupled to the phosphorylation of ADP. Respiration proceed only when ADP is present. The amount of oxygen consumed depend upon the amount of ADP added. Okay, so there is a series of sequential oxidation and reduction reaction during this electron transport chain, and what is the final acceptor of those electrons? Oxygen act as a final acceptor. Okay, so the electron pass from NADH or FADH2 to oxygen, and this, after accepting those electron, this oxygen is mainly responsible to produce water through a series of protein complex, and this water is also known as metabolic water, which is generated by this during this electron transport chain. Since NAD and FAD are in limited supply, so they must be recycled during this process. And this is the electron transport chain. This is the inner membrane of mitochondria, and this electron transport chain contains so many carrier molecule, as you can see over here, as a complex 1, complex 2, complex 3, complex 4. Some molecules act as a mobile carrier like cytochrome C. Okay, so they act as a mobile carrier. So as this electrons from NAD produced in the mitochondrial matrix during citric acid cycle, just now we have discussed citric acid cycle, there are certain steps where NADH has been generated, FADH2 has been generated. So electrons are produced from NADH produced in the mitochondrial matrix during citric acid cycle, are oxidized by NADH dehydrogenase in complex 1. This is complex 1, NADH dehydrogenases. Here conversion of NADH to NAD and electrons, and these electrons then pass through ubiquinone to second complex, to third complex, to mobile carrier, that is cytochrome, to final complex 4. Okay, so these carrier molecule actually act as a mediator molecule which help to transfer this electron from one molecule to another molecule and then towards the final acceptor, that is oxygen. Clear? This ubiquinone, this Q, ubiquinone also receive reducing equivalent via FADH2. As you can see, this is FADH2 present over here. This electron has been accepted by from complex 2, electron has been accepted by this ubiquinone. This reduced ubiquinone is then oxidized with the transfer of electron to cytochrome C. This further transfer the electron to cytochrome C via third complex, means cytochrome BC1 complex. This BC1 complex. Okay, so all the electrons from complex 1 to complex 2 to complex 3, they all transfer their electrons from different carrier molecule to cytochrome C, and cytochrome C act as a mobile carrier which further transfer these electrons to complex 4, where oxygen is present, which act as a final acceptor of those electrons. It actually accept all the electrons and utilizing some proton which already present in this particular part, and further one electron one proton combine together and produce hydrogen and convert this oxygen into water. Clear? Now this cytochrome C, this one. Here we are talking about this cytochrome C. It is a small protein attached to the outer surface of the inner membrane. As you can see over here, this is the inner membrane and this is the outer surface. So that's why it is written over here, this cytochrome C is a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for transfer of electron between complex 3 to complex 4. This is complex 3 and this is complex 4. So this cytochrome C act as a mobile carrier and act as a mobile carrier for the transfer of electron from complex 3 to complex 4. And this complex 4 refers to as cytochrome C oxidase complex containing cytochrome A3, cytochrome A3 and 2 copper centers, because copper ion act as a copper C plus as a this form and this form. In the both ionic form they act as a stable molecule. Similarly, iron in initial complex 1, 2 and 3, iron act as a main center molecule, because iron can be present in a +2 or +3 form, ionic form and act as a stable molecule. So they can receive, if I am iron +3 form, then I can receive an electron and convert myself into iron +2, and then I can transfer this electron to the next carrier molecule and then further myself into +2 to +3 form, and in the both the ionic form I am act as a stable molecule, stable ionic form, sorry. So that's why in initial these molecules, iron act as a main center molecule as a FES form, and in the last complex 4, copper ion act as a main molecule, center molecule, for act as a transferring. They utilize iron and copper for to transfer the electron from one form to another form, one carrier molecule to another carrier molecule. Okay, when electron pass from one carrier to another via complex 1 to 4, from 1 to 4, in the electron transport chain, they are coupled to ATP synthase, which act as a complex 5. Finally, the electrons move from this complex to 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 this complex. Here there is a fifth molecule which is known as F0F1 particle, which is known as complex 5. And here ATP synthase enzyme is present. Okay, and here ATP, ADP plus inorganic phosphate is present, and in the presence of this ATP synthase, it will mainly responsible for the production of ATP. How this happen? Let's see. The number of ATP molecule synthesized actually depends upon the nature of electron donor. Oxidation of one molecule of NADH give rise 3 molecule of ATP. Just now we have discussed this, and one molecule of FADH2 produce 2 molecule of ATP. Right? Although the aerobic process of respiration takes place only in the presence of oxygen. The role of oxygen is limited to the terminal stage of process, yet in the presence of oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as a final hydrogen acceptor, or we can say final electron acceptor, because that hydrogen molecule is having one electron and one proton, right? So electron pass from one carrier to another carrier molecules. As you can see over here in this particular picture, these electrons are passed from one carrier first to 3 to 2 to 4, like this, one complex to another complex, and all the protons are passed from matrix to inner membrane space, because there is no need of those protons, so the concentration of proton gets higher and higher on the inner membrane space. Okay, and the concentration of proton gets decreased down in the matrix, because the remaining proton which are present over here, and this has been utilized by the oxygen, as this electrons which pass from one complex to another complex and finally accepted by this oxygen. So by utilizing protons, remaining protons, and accepting all the electrons from here, this oxygen gets converted into water. It will produce water. That's why it is written over here, oxygen act as a final hydrogen acceptor or final oxygen acceptor. And the concentration of proton decrease over here in the matrix and increase in the inner membrane space. So what happened? There is a proton gradient develop. Proton gradient develop. So because of the proton gradient, in a very fast way, all the protons very quickly, in a very energetic way, they move from F0 F1 particle. So in the presence of ATP synthase, during this time, this ADP and inorganic phosphate combine together and make a bond and produce this ATP. This is known as oxidative phosphorylation, production of ATP. Clear? So just see this diagram again. This complex one, this one is the complex one. Okay, this complex one where NADH electron enter into the chain. NADH electron enter into the chain, and as already I told you, this is iron is present in complex 1, complex 3 and complex 2. Already I told you, initial 1, 2, 3, all three complex contain iron sulfur, because it is easily converted into, it's both the ionic form, +3 and +2 form. Okay, so electron first move from FMN, flavin mononucleotide, to ubiquinone, which acts as a ubiquinone to ubiquinol form, and then third, which is having cytochrome C BC1 complex, and then both the complex 2 and complex 1 push their electrons through ubiquinone to complex 3, where this mobile electron carrier means cytochrome C collect all the electrons from complex 3 and pass it to complex 4, where cytochrome A and A3 act as a carrier and then push and give this all electrons to this oxygen, and this oxygen further utilize the proton which is remaining proton which is present in the matrix and electrons and convert itself into the water. Okay, so complex 1 where NADH electron enter into the chain, complex 2, this is the complex 2, where FADH, you can see FADH, electron enter into the chain, electron pass from 1 and 2 to coenzyme Q ubiquinone, complex 3 shuttle electrons to cytochrome C, complex 3 to cytochrome C, already I told you, and then cytochrome C shuttle electrons to complex 4. Shuttle electrons to complex 4. ठीक है. So this is how movement of electron occur from 1 to 3 to 2 and 4 and like this. This is the main pattern, and all the protons, they just push towards intermembrane space, and that's why the concentration of proton continuously decrease in the matrix and increase in the intermembrane space. And because of the change in the concentration of proton, there is a gradient has been developed, which is known as proton gradient. So with a very fast way, this proton move from inner membrane space towards the matrix side through F0 F1 particle, and this F0 F1 particle contain ATP synthase, and in the presence of ATP synthase, this ADP and inorganic phosphate combine together and produce ATP. Okay, so this is oxidative phosphorylation. So this is a sequential process, orderly process, in which on 3 different steps ATP has been produced. First step, glycolysis, which occur in the cytoplasm. You can see this is the cytoplasm where glucose get converted into pyruvate and ATP has been produced. Little bit ATP has been produced. Okay, and then this pyruvate enter into the mitochondria where pyruvate oxidation occur. It will convert into first acetyl coenzyme, and then that acetyl coenzyme combined with oxaloacetic acid and produce citric acid, which is the first product of this Krebs cycle or citric acid cycle, and produce little bit ATP over here also. These two known as substrate level phosphorylation. And during citric acid cycle, lot of NADH, FADH2 has been produced, which is further utilized by the electron transport chain, where so many carrier molecules are there present on the membrane, and they pass electron from one carrier to another carrier to another carrier and push all the protons towards the out intermembrane space. And because of the push, there is a gradient has been developed, and then because of the gradient, immediately what happened, that proton come with a very forceful way through F0 F1 particle inside the matrix, and then that time huge amount of ATP has been produced, and this is known as oxidative phosphorylation. And at this stage also ATP electrons has been used up or finally accepted by the oxygen, and production of H2O also occur, which is known as metabolic water. So there are 3 steps in which ATP has been produced. Okay, so this is a sequential step, orderly pathway in which one substrate forming the next with glycolysis, TCA cycle, and ETS, electron transport system pathway, following one after another and produce ATP at each level. Okay, so this is all about the oxidative phosphorylation, electron transport chain, citric acid cycle. Now, if all the concept has been clear, then it's okay. If you have any doubt, you need some kind of clarity, you can write down to me in comment box. We will discuss it later on. Till then, take care. Bye bye. Keep learning. Namaskar.